So you have just finished the exploration activity, Genetics in the Name of Love. Let's just review briefly the main ideas that you should have taken away from the activity. If you remember, in this activity we follow a fictional couple through an awkward dilemma. Jack and Jill would love to get married, but Donald, Jack's father, was an anonymous sperm donor, and Jill was the result of an anonymous sperm donation. Since they're from the same town, it's possible that Donald is both Jack and Jill's father. Yikes. To begin, we first had to discover the process of meiosis in order to understand how genes are passed on to offspring. Remember that each individual contains 2 complete sets of their chromosomes, or 23 homologous pairs. I have pictured here a real human karyotype with all 23 pairs laid out, and here is a pictorial version of our activity. So this is Donald, or Jack's father, and we have just depicted 4 of his chromosome pairs here. So he has a number 1 from his mom, and a number 1 from his dad, a number 9 from his mom, a number 9 from his dad, and so on. Now, homologous means that chromosomes carry the same genes in the same order, but not necessarily the same alleles or versions of the gene. So this chromosome here could have a big A for a particular gene, where this chromosome has a little a for that same gene. Having 2 copies of each chromosome makes human beings diploid, or 2N. Because humans participate in sexual reproduction, which is the combining of DNA from 2 individuals, they must create gametes, which is just a fancy name for egg and sperm, and these gametes are haploid, or 1N, meaning that they only contain 1 copy of each chromosome. When these 2 gametes recombine, the resulting offspring is back to being diploid. Let me take a moment to explain what N means. N indicates the number of unique chromosomes. So, if an organism has a diploid number of 14, or 2N equals 14, this means that they have 2 copies of 7 unique chromosomes. So they've got a number 1 from mom and a number 1 from dad, a number 2 from mom, 2 from dad, 3 from mom, 3 from dad, 4 from mom, 4 from dad, 5 from mom, 5 from dad, 6 from mom, 6 from dad, 7 from mom, 7 from dad. Alternatively, if I tell you that an organism is 8N equals 32, you should be able to conclude that this organism has 8 copies of 4 unique chromosomes. In other words, it's octoploid. So it has 8 number 1s, 8 number 2s, 8 number 3s, and 8 number 4s. Diploid cells in humans make haploid gametes through a process called meiosis. I will demonstrate that briefly right here. The first phase of meiosis, or meiosis 1, begins with prophase 1. Now I'm just going to do 2 chromosomes here. So let's say we have chromosome number 1 from mom and chromosome number 2 from mom. And we also have chromosome number 1 from dad and chromosome number 2 from dad. Now notice in prophase 1 that I've drawn them very close together, because it's during this time that big sections of them can swap places in a process called crossing over. I'm not going to draw it here because it's complicated to draw, but suffice it to say we get a lot of genetic mix-up during prophase 1. During metaphase 1, the chromosomes will line up along the middle of the cell as homologous pairs. So I'm going to draw that here. We'll draw number 1 here and number 2 here. Now notice that I'm drawing the blue on the bottom here and the blue on the top here, because it doesn't matter. This is where the law of independent assortment gets fulfilled, that these chromosomes can assort from one another independently. We're also going to get these spindle fibers that are going to grow out from the edges of the cell to try to pull these chromosomes apart. It's during anaphase that these chromosomes are actually pulled apart. So I'm going to draw that here. We've got number 1 from mom going up and number 2 from mom going down. And the reverse, number 1 from dad going down and number 2 from dad going up. And again, I'll draw the little spindle fibers that are pulling them up. During telophase, we're getting a little bit of pinching of the cell, so it'd be more accurate to draw it like this. And again, we've got this chromosome in here, this chromosome in here, and this chromosome and this chromosome. And then we have cytokinesis, where the cell splits into 2. So now starts the process of meiosis II. Now let's point out here in prophase 2 that we have our two cells and that they each contain only one number 1 and one number 2. So this actually makes them haploid cells at this point, or 1N. Alright, during metaphase 2, these are going to line up along the center plate, just like we see during mitosis. So here we're going to line them up like so. Number 1 and number 2, we're going to get spindle fibers that'll grow out on each side to pull these apart. During anaphase 2, we are just simply going to pull those sister chromatids, or duplicate copies of each chromosome, apart. So we'll draw that here. This is the one side, this is the other side, pulling apart. And the same with the blue chromosomes. And I'll draw our spindle fibers coming up. Ooh, that's a little sloppy, sorry about that. Okay, and during telophase 2, again, it would be more appropriate to draw them sort of pinching already. So we'll draw that like so. And we'll draw our chromosomes in there. So we have mom's number 1 and dad's number 2 in these two copies. And in the bottom ones, we have dad's number 1 and dad's number 2. Then cytokinesis happens once again, dividing them into 4 haploid cells. So you can see here in the 4 resulting gametes, the top 2 have mom's number 1 and dad's number 2, and the bottom 2 have dad's number 1 and mom's number 2. Now keep in mind, if crossing over had occurred, then 2 of these gametes would actually have swapped information, and all 4 would be genetically unique haploid cells. The first inheritance pattern is called complete dominance, also known as complete autosomal dominance, and autosome is any chromosome other than the sex chromosomes. Now, for this type of inheritance pattern, it means that one allele is completely dominant over the other allele. So we have 3 potential genotypes. We could be big A, or homozygous dominant. We could be heterozygous, big A, or we could be homozygous recessive, little a. Since one is completely dominant over the other, if you are homozygous dominant, your associated phenotype is whatever the dominant phenotype is. Likewise, with your heterozygote individuals, they will also show the dominant phenotype, but a homozygous recessive individual will show the recessive phenotype. So let's illustrate this in some way. If the dominant color, let's say for hair, is black, then if you are homozygous dominant, you will have black hair. If you're heterozygous, you'll also have black hair. But if you're homozygous recessive, you'll be lacking that black pigment, and you'll have white hair. Now let's look at codominance. In codominance, neither allele is dominant over the other. In fact, they're both dominant. So normally you would use different letters, but I'm going to use the same letters and colors just to have it easier to understand. So we'll do the same 3 genotypes. You're either 2 big A's, a big A and a little a, or 2 little a's. In this case, if you have 2 big A's, you're going to show whatever that big A phenotype is. In this case, we had it as black. And if you have 2 little a's, you'll show whatever that little a phenotype is. Notice I'm not saying dominant or recessive, because neither one's dominant or recessive. But if you have a big A and a little a, you'll actually show both a big A and a little a phenotype. So in this case, we'll call the black hair as big A and the white hair as little a. But someone who is heterozygous might be black and white polka dotted. The next inheritance pattern is incomplete dominance. In this case, neither allele is dominant over the other. So again, the 3 genotypes we could have is homozygous dominant, heterozygous, and homozygous recessive. The homozygous dominant shows the dominant phenotype. The homozygous recessive shows the recessive phenotype. But in this case, the heterozygote, instead of showing both alleles, shows an intermediate or mix between the 2. So if I were to illustrate this, we'd have our dominant black hair, our recessive white hair, and then the heterozygote would be some kind of mix between, or a gray. Okay, and then the last inheritance pattern is sex linkage, specifically X-linked traits. So these are traits that reside on the X chromosome. So for a girl, since we have 2 X chromosomes, you can be homozygous dominant, you can be heterozygous, and notice I put them up on the X chromosome to indicate that they're on the X chromosome, and you could be homozygous recessive. So it works the same way as before. You would show the dominant phenotype, or the recessive phenotype. But in a male, because he only has one X chromosome, there are only two possible genotypes. He either has the dominant on his X, or he has the recessive on his X. And again, these show the same way. You're either dominant, or recessive. But again, for a male, he can't be heterozygote. Throughout this activity, you learned how to cross gametes in an organized fashion called a Punnett square to determine potential offspring. I'm just going to demonstrate one here. Now remember, you should always think of Punnett squares in terms of crossing gametes, or eggs and sperm. So I always draw the egg and sperm on each side of the square. So let's say that Donald was heterozygous for some allele, and that Daisy was homozygous recessive. So those would be the gametes they could produce. And then you simply write them in the squares. So one of their offspring potentially could be heterozygous. The offspring resulting from the other two would be homozygous recessive. Again, a heterozygote offspring and a homozygous recessive offspring. It's as easy as that. The last thing you encountered were linked genes. This means that the genes were on the same chromosome, and in this case they won't assort independently. So let's look at two scenarios to try to understand this better. In this first scenario here, we're looking at two genes, gene A and gene B. Gene A happens to exist on chromosome number 1, and this individual is heterozygous, and gene B is on a separate chromosome, or chromosome 2, and they're also heterozygous. So if we were to look at the potential gametes this individual can make, he could make a big A and a little b if I chose both gray chromosomes, a little a and a big B if I chose both blue chromosomes, and then the two other combinations are big A and big B if I chose a gray and a blue, or little a and little b if I chose a blue and a gray. So you can see that there are 4 potential gametes here, and they're all assorting independently from one another. In this second scenario, you can see that gene A and gene B are both on the same chromosome number 1. So in this case, if we were to look at the potential gametes, we can either grab the gray chromosome and get big A and little b, or we can grab the blue chromosome and get little a and big B. There's no chance of getting these other two gamete types, and so you limit the amount of offspring, or the different varieties of offspring that you would get. So anytime you get less than what you would expect in the variety of offspring, you should suspect that the genes are linked. Now keep in mind that we could have a crossover event between these homologous chromosomes, and if that crossover event happens anywhere between the two genes, such that they switch places, then you can get these recombinant gametes and end up with offspring that you wouldn't necessarily expect given that the genes are linked. The more often that this occurs, the farther apart those genes are on the chromosome. That concludes this tutorial session and should give you an overview of everything you should have learned from the Genetics in the Name of Love exploration activity.